Uh, Good morning to all of you. In the last class, we have tried to understand about the antigen and the structure of the antibody. So today, we will move forward with the cells of immune system because antibody and antigens are complementary to each other. And now, when we talk about the antibodies, especially which are produced against the infection or the specific antigen, there are how they are formed, what are their roles, all those things we have to discuss here, and what are the different types of antibodies or what is known as the lymphocytes. So all these things we will discuss under the heading the cells of the immune system. I hope all of you can see clearly. Kanata idea slide.
Yes, sir.
Okay.
Yes, sir.
Now, in our main defense system of the body, if you take, there are certain things apart from these cells. First one is the skin. Skin is a physical barrier for the entry of any of these antigens or pathogens. Suppose most of the time the pathogen enters our body not through the skin. Uh, they mostly enter inside the body either through mouth, nasal, or through eyes or ears or various openings that we have. Or sometimes even the pathogens may gain entry in our body if there are lesions, cuts, injuries in the skin. Otherwise, skin is a protective barrier to all the living beings. Uh, then the another defense is the glands and their secretion. There are many glands and they secrete uh, certain chemicals. For example, suppose if something enters in your eye, immediately there will be tears which is secreted by the lacrimal glands. Like that, in the ear, again the ear wax. Uh, so these are certain secretions which will, all of them are antiseptic in nature and they will prevent the entry of these pathogens in the body. After crossing either the skin or the effect of these glands and their secretion, suppose the pathogen enters our body, the white blood cells or leukocytes, which are popularly called as lymphocytes, they are responsible for tackling or handling those pathogens which have gained entry into the body. So, when I talk about the lymphocytes, basically there are two types. One is B lymphocytes and another one is the T lymphocytes. Actually, B lymphocytes are having the function of antibody production, which you have already seen. The T lymphocytes mainly they are self-activation. They are concerned with self-mediated immunity. All these, irrespective of whether it is a B lymphocyte or a T lymphocyte, they take birth in the bone marrow. Bone marrow is especially in the bone marrow of some larger bones, where that you can see some of the cells, what you call the protoplast cells, they are not differentiated. Stem cells, they are also called stem cells. Uh, these stem cells of bone marrow, there's a small spelling mistake, please excuse me for that bone spelling. The stem cells are produced in our body, especially in the bone marrow. And these stem cells then differentiated into two different types of cells or two different series. One is lymphoid series and another one is myeloid series. Myeloid series, it has other white blood cells like macrophages, dendritic cells, eosinophils, mast cells. They are directly attacking on the pathogens. Whereas the lymphoid series, we have basically two types. One is B lymphocytes and T lymphocyte, and some consider even natural killer cells, NK cells in short, they are called, also the lymphoid series. So, irrespective of whether it's a B lymphocyte or T lymphocyte, they originate in the bone marrow. What do you mean by B lymphocyte? Many consider the B lymphocyte as bone marrow lymphocyte. No, actually, B stands for bursa fabricius lymphocytes, and T lymphocytes, the thymus derived lymphocytes. T lymphocytes are the thymus derived lymphocytes. B lymphocytes are bursa fabricius derived lymphocytes. Though we do not have bursa fabricius, that is found only in the birds. I will take it later. Mm, then another one is the natural killer cell. So here in the next slide you will see how they are produced. So the red bone marrow, especially the red bone marrow, that produces uh, these stem cells. These stem cells will mature into either pre T cells or mature B cells. The B cells or B lymphocytes mature in the bone marrow only. But whereas these T lymphocytes, they are born as a stem cell in the red bone marrow, then they reach the thymus. In the thymus, they are trained and educated, and then it becomes T lymphocytes. As far as the origin of both B lymphocytes and T lymphocytes are concerned, they are same. Some of them some of them mature in the bone marrow and they become mature B lymphocytes, whereas some of the stem cells they migrate to the thymus gland, where these our thymus gland educate and train them and they become uh, T lymphocytes. Now there are two ways and they are interconnected also that you have to see. The mature B cells they they are developed into the plasma cells or antibody cells because when the plasma mature B cells, B lymphocytes, they have antigen receptors on it. When the antigen binds on the receptor site of the B lymphocyte, the activation of the B lymphocyte or B cell takes place. This activation of the B cell change produces the plasma cells. These plasma cells produces what is known as the antibodies. Whereas if you come to the thymus derived T cells or thymus derived lymphocytes or T lymphocytes, they mature in the thymus and when they mature, they develop into either CD8 or CD8 or CD4 cells, and some of them mature into helper T cells. And these helper T cells also help the activation of B lymphocytes to change themselves into plasma cells. Otherwise, these helper T cells also help in the activation of the T cells, and they develop into CD8 cells, and that is killer T cells, and which are responsible for cell mediated immune responses. So now first, let's concentrate on the B cell or B lymphocyte. So these B lymphocytes are formed within the bone marrow, developed. They take birth in the bone marrow and undergo their development also within the bone marrow. So they do not migrate to any other gland. Instead of that, they remain in the bone marrow and develop there. And these B lymphocytes have four important functions. One is the B lymphocytes can interact with the epitopes of the antigen. That's the binding sites of the antigen because on these B lymphocytes there are immunoglobulin receptors. And on these immunoglobulin receptors, they bind with the antigenic epitopes. And once they bind with the antigenic epitopes, that develops, this B lymphocytes develops into plasma cells. And once they are developed into plasma cells, these plasma cells will release large amount of specific antibodies, specific in the sense specific to the antigen from which it has been formed. That is one thing. Or otherwise, they will circulate as memory cells. Some of them will secrete large amount of specific antibodies. and antibodies will take care of the antigens and they will nullify the effect of antigen. Whereas few of them, they circulate in the blood as a memory cells. The role of the memory cell is to uh, identify the antigen. Suppose if it reinfects the same person, already now these memory cells know the type of the antigen, because of that antigen only these memory cells have been formed. Once those antigens again infect us, immediately the memory cells will recognize them and start producing more and more antibodies. Then apart from that, the B lymphocytes, they also sometimes present this antigenic peptides to T lymphocytes, because the T lymphocytes initiate the cell-mediated immunity. The T cells will directly engulf or destroy these antigenic peptides. Uh, uh, So this is the role of the B lymphocytes. So they see here the B lymphocytes, they become plasma cells, and once they become plasma cells, they produce the antibodies. When where only when the B lymphocyte binds with the antigen, it turns into plasma cells, and the plasma cells will go on producing the antibody and it triggers the immune response. This is known as the initiation of the immune response or triggering or activating the immune response. Uh, as a result of which, what happens? The plasma cells will be able to produce large number of these antibodies. And this B lymphocytes, they are basically stem cells which have differentiated into B cells. And a very interesting feature of the B lymphocyte, the B lymphocytes, when they mature, they differentiate into immunocompetent B cells. When they mature, they differentiate into immunocompetent B cells with a single antigen specificity. Remember, a particular B cell always sensitive to a specific single antigen. If there are different types of antigen, there must be different kinds of B cells, immunopotent, immunocompetent B cells. A single immunocompetent B cell means it is specific to single antigen. If there are different antigen, there will be different B cells formation will take place. So this especially if you look into the immature B cells, these immature B cells usually have high affinity receptors for self antigen and die or fail to mature. This is sometimes it happens. Immature B cells, some of them express very high affinity for the self antigen. Self antigen means the protein of our own body, and as a result there will be immune response and they will die or fail to mature and they are taken out. This process induces the self-tolerance and reduces the autoimmune disease. Otherwise, what would have happened if these uh, some of the B cells which express high affinity for self-antigens, if they do not die or fail to mature, they would have triggered uh, immune response against our own body, which is called as autoimmune diseases like psoriasis and all. So this is mostly prevented because the B cells which are having high affinity toward the self antigen, they die or they fail to mature. So these immature B cells, if you look into that, they express immunoglobulin M receptors on the surface. Whereas mature B cells, they have immunoglobulin M and immunoglobulin D molecules on their surface. Uh, Both immunoglobulin M and immunoglobulin D molecules, they are nothing but the receptors for the antigen, which are found on the surface of the B lymphocyte. On the B lymphocytes, there are receptors, and these receptors for antigen, and these receptors are known as immunoglobulin M and immunoglobulin D. Whereas the other type of B cells, as I have told you, the memory B cells, they express immunoglobulin G and or immunoglobulin A or immunoglobulin E on their surface. Just don't get confused here. Uh, Immature B lymphocytes have immunoglobulin M as receptors on their surface. Immunoglobulin B cells, they have immunoglobulin M and immunoglobulin D molecules on their surface, and they are nothing but the receptors for specific antigen. And immunoglobulin N and D, both of them serve as a receptor for antigen. When the foreign antigen comes, uh, they bind with the epitope of immunoglobulin of the B cell, that is immunoglobulin M or immunoglobulin D. Then there is other kind of cells which I have already told you, the memory B cells. On the surface of the memory B cells, you can see immunoglobulin G or A or E on their surface, and uh, they will recognize the subsequent antigens if they happen to reinfect the same person or same individual. They can recognize immunoglobulin G, A, and E. They can recognize only those antigens which have previously infected the individual, because they keep a memory, they keep a storage of memory, and they can immediately target those already in experience in tackling the immunoglobulin, and they can identify the fresh infection of the old antigen. The B cell receptors, um, the receptors have Fc portion on immunoglobulin G and receptor for C3 component, the complement. So these B3 B cells, uh, they have immunoglobulin M and D, and they have an uh, Fc portion of Ig and a receptor for C3 component of the antigen. Here usually they express a array of molecules on the surface that are important in B cell interaction with other cells. Uh, this portion of Fc portion on immunoglobulin G and becoming a receptor for C3 component is an important as far as the B cell interaction with other cells are concerned, especially MHC2. MHC2 means major histocompatibility complexes, or CD4, B7. These are different uh, immune cells. Now, we should know the mechanism of immunity, humoral immunity. Antibody induces resistance. Humoral immunity through the antibodies. And we should know that um, how these antibodies can induce the humoral immunity. In fact, the humoral immunity, what we call in any individual, is because of the antibodies. Antitoxins neutralize the bacterial toxins, because these antibodies will have antitoxins, whereas the antigens will have the toxin. Antigens are nothing but the toxin, and these antitoxins of the antibodies will neutralize this bacterial toxin, especially in diphtheria and tetanus. This is happening. Now you may ask how these antitoxins are developed. Antitoxins you can uh, develop because of three reasons. One is previous infection. If at all there's a previous infection, then immediately the memory cells will have the record of those infection, and immediately it will start producing the antitoxin. So that's the reason. Suppose if you have got the chicken pox again, people say that, and you also agree that you will never catch the chicken pox again, or this is because of the previous infection. So that's why usually a human being will get chickenpox once in his lifetime, because the previous infection memory cells are there and they will immediately produce antitoxins. Or sometimes the next way of developing the antitoxin is artificial immunization. What is artificial immunization, mostly it is the vaccination. You are all vaccinated against BCG, MMR, there is the mumps, measles and rubella, or polio. How? So when artificially we have introduced those antigens, that's attenuated. Most of them are attenuated vaccines, and when they are injected, our body will produce antibody and keep the memory cells ready. So this is because of artificial immunization. Then sometimes it is transferred passively as anti-serum. Uh, that is what we call nowadays the, you must have read in plasma therapy, which is very popular even for treating this COVID-19. This is transferred passively as antigen. Uh, Suppose if you have already infected, then our body may take lot of time to produce the antibody. So that's why what is done is some of the anti-serum is transferred already from a person who is immune to that disease. Who is immune to that disease. Uh, a good example is, well, suppose if there's a tetanus, then usually they transfer this anti-serum. Or another very interesting thing is anti-serum which have the ready-made antibodies. In the snake bite, when there's a snake bite, what we get is antitoxin, what we call. This antitoxin is nothing but the ready-made antibodies. So the antibodies are produced in the horse blood, and then antibodies are separated, and those antibodies are injected immediately because there is no time for our body to produce the antibody. So that's why the ready-made antibodies are transferred. So these are how these antitoxins are produced and effective in this one. Uh, so as a result, it, it, uh, these antitoxins neutralizes the toxin and thus prevents combination with the tissue cells. It will not allow the antigen to enter into the tissue cells. Before that only they are tackled and finished. So this is one way. And another one is antibodies. Suppose or when the antigen enters the body, how antibodies will fight against them. So first, antibodies attach to the surface of the bacteria or any pathogen, and when they attach, it acts as an uh, opsonins and enhances the phagocytosis. That's a cell-eating process. First, it will try to engulf, eat the entire antigen and neutralize it. Then, uh, at the same time, it will prevent the attachment of the microorganisms to their target cells because each antigen has a target cell. For example, uh, a bacteria will affect only the gut. Then what happens? The immunoglobulin A, which is on the surface of the antibodies, will prevent the attachment of those microorganisms to their target cells in the gut. Then, meanwhile, it will also activate complement and lead a bacterial lysis. Then immediately activate the complement cells, mast cells, plasma cells, and other things to, and they engulf the bacteria by phagocytosis and lysis because inside there is a lysosome and they can break it. Then some even sometimes, if there are more number of bacteria, they want to make a clump of bacteria, agglutination process. The all the bacteria get agglutinated and then they are phagocytized. That means they are tackled by cell eating process. So this is the mechanism. So here, when the antigen, this is the flow chart, when the antigen binds on the surface of the B cell, because there's a specific antigen, there's a specific B cell, and they attach them. And when they attach, the antigen is taken inside, and the antigen is inside the B cell, they are processed. And especially the class 2 MHC, the major histocompatibility uh, complexes, and uh, they are processed and displayed. Then what happens? That there are activated helper T cells. They will help these antibodies to produce the plasma cells, and these plasma cells now produce the antibodies again. The antibodies will uh, neutralize this one as well as start producing more number of antibody. This is how any pathogen that gains entry into the body of the host is tackled. So this is how the B lymphocytes function. Then coming to the T lymphocytes. T lymphocytes also originate in the bone marrow. Like the B lymphocytes, they also originate in the bone marrow as stem cell. But after uh, taking birth in the bone marrow, they do not develop that. Instead of that, some of these T cells migrate to the thymus. And in the when they migrate to the thymus and enter the thymus, in the thymus, especially in the outer cortex of the thymus, the T lymphocytes start acquiring specific receptors. So that's why they are called thymus derived lymphocytes. T lymphocytes means thymus derived lymphocytes. They acquire specific receptors, that is T cell receptors. These receptors are known as TCRs or T cell receptors. These receptors make the lymphocyte into a single antigen specificity. Again, these T cells also show single antigen specificity. It all depends upon the specific receptors that are found on the T lymphocyte. For this, in the thymus, the T cells are trained and educated. And these T lymphocytes, they acquire specific T cell receptors on their surface, and these specific T cell receptors make the lymphocyte to a single specificity. It can tackle single pathogen, single group of pathogen. And then suppose if the T cells come in contact with the pathogen, then it respond immediately. How they respond? They respond immediately by proliferation and producing clonal cells. Clonal cells mean exact copies of its own. It goes on producing clone cells or exact copies of this one. And these clones may differentiate to express CD3 cells, CD4 cells, or sometimes CD8 cells. Different types of T cell receptors. Clones are formed. So T lymphocytes, most of the time, they become CD4 cells. So CD4 cells are also called as helper T cells, or sometimes some of them change into CD8 plus cells, which in turn can become killer T cells. The CD8 cells are killer, known as killer T cells, also called as cytotoxic T cells, because they can directly kill the pathogen. But whereas helper T cells will help the B lymphocytes to produce antibodies, but whereas here the CD8 cells, they themselves become killer T cells, and they kill the pathogen on their own without the aid of the antibody. So that's why they are also called as cytotoxic T cells. This this is everything is happening in the uh, cortex of the thymus. Now, in the medulla of the thymus, these T cell receptors, they can recognize the major histocompatibility molecules loaded with normal self peptides. They recognize that these MHC proteins. MHC is a class of protein. MHC, the major histocompatibility molecules. So they are loaded with normal self peptides. It is known as PMHC, and these T cell receptors are capable of binding with low affinity to this PMHC. That is the self peptides, and it will receive positive selection, but uh, though they have the low affinity of binding to this PMHC, uh, it will receive some positive selection signals to divide and establish the groups. So when they are loaded with normal cell peptides, these T cell TCRs, that the T cell receptors, they will get some signal and start producing their clones. Now, uh, this TCR sometimes they can bind too strongly with PMHC also, and they go negative selection. And this selection process will eliminate the potentially most harmful reactive T cell. Why? What is the role? This selection is very, very important process. It will This selection process will eliminate some of the most harmful self-reactive T cells. Otherwise, these T cells will react with the antigens or the proteins of our own body. So that's why this selection process, especially the negative selection process, will eliminate the potentially most harmful self-reactive T cells. This is a central self-tolerance, and thus protect us from autoimmune diseases. Then, once the T cells becomes mature, they can do all those things. But whereas the immature T cells generally express themselves either into CD4 cells or CD8 cells. I have told you, CD8 cells they can directly tackle. CD4 cells they function as helper cells, and as they mature, the T cells with T cell receptors that have the affinity to bind with major histocompatibility proteins or MHC class II protein, and they become helper T cells with CD4 molecules only, whereas CD8 cells which have the affinity to bind with MHC class 1, if they have the affinity for MHC class 2, they will develop into CD4 cells. If they have affinity to bind with MHC class 1, they will become cytotoxic T cells with CD8 molecules only. In mature, when they mature, they are positively selected T cells are MHC restricted. All cells, all mature T cells do not show affinity for these major histocompatibility proteins. Many times they are restricted because the CD4 T cells are MHC2 restricted because they recognizes the specific peptide only, and when they are presented in association with MHC class 2, they do not uh, identify the MHC class 2, whereas CD8 cells uh, are MHC class 1 restricted, and they recognize self-protein peptides only when they are presented in association with specific MHC1 molecule. Uh, so this, as a result, you can see uh, the CD4 cells will not match with MHC2 complex, and CD8 cells will not match with MHC1 molecules. These CD4 cells, I have told you, they are also called CD4 T helper lymphocytes. Helper lymphocytes or TH cells. These TH lymphocytes, usually they recognize the antigen on the surface of uh, APC, that is antigen uh, presenting complex. APC, antigen presenting complex. Uh, this TH lymphocytes or helper lymphocytes, they recognize the antigen on the surface of antigen presenting complexes or antigen presenting cells, we can say that. Antigen presenting cells are nothing but the antigens only where the antigen is binded with a normal cell. We call it as antigen presenting cell. Now the our body cell is having an antigen because on the surface of that the antigen has attached. So this antigen presenting cells in association with the class 2 MHC molecule, the helper T cells recognize the antigen. Once the helper lymphocyte recognizes this antigen, they get activated and secretes several cytokines. They secrete several cytokines. Uh, there are two main subsets, Th1 and Th2, and mainly this subset differentiation is based on the cytokine they produce. What type of cytokine they produce? This Th1 usually produces cytokine of CM1, and usually it causes the inflammation. When there's an infection, there's an inflammation. Suppose if you have a cut, some pathogens have entered, bacteria and other things. There will be inflammation. This helper T cells, that the Th1 cells, they produce cytokinase of CM1, and as a result, there will be inflammation. Whereas the helper 2 cells produce mainly that helper cells. It also produce cytokine which produce the B cell, and also produces suppressor cytokines. The helper T helper T2 cells, they also produce cytokine, but that cytokine will produce stimulate B cells. But whereas in the first case, helper T1 cells, they produce cytokine kinds of CM1, and it causes the inflammation. Whereas cytokine 2, the helper cell 2 produces the or stimulate the B cell. Not only stimulate the B cell, it also produces suppressor cytokines. I will come to that. What is the role of this suppressor? So first thing is, the function of the helper cells is stimulate the cytotoxic and suppressor T cells. Suppressor T cells are also very, very important. One is they stimulate cytotoxic cells as well as suppressor T cells. Second thing is stimulate the growth of B cell, and also uh, stimulate for the differentiation to form plasma cells. Then it also activates our macrophage system, macrophages, and even they get feedback stimulatory effect on helper T cells themselves. They get the feedback. What should be the B side, amount of B sides, what should be the suppressor cells, all those things it will get. So see here, helper T cell activation. So this is the antigen recognition. This is antigen presenting cell. This is the antigen. This entire cell is associated with this antigen, and this is when it is presented to MSG2, they produce CD4 cells, and they are the helper T cells. And this, or some of them develop into effector cells. Uh, they have the clonal selection. The clonal selection, two types of killer clones. Uh, some develop into memory T cells, some develop into neutrophils, macrophages, or killer T cells or B cells. B cells are for humoral immunity, and these killer T cells, they directly kill that one, the pathogens. Whereas neutrophils and macrophages, they also phagocytize the uh, antigens, especially the CD8 cytotoxic T lymphocytes. Uh, they are present to constitute about 35% of peripheral T cells. 35% of peripheral T cells are CD8 cells. So this CDLs recognizes that the CTLs, the cytotoxic T lymphocytes, see, cytotoxic T lymphocytes, they recognize the antigen on the surface of the target cell in association with MHC class 1 protein. Then they are activated and kill the virus infected cell, or even the tumor cells are also targeted by this way. Whereas the cytotoxic T cells, especially the CD8 cells, killer cells, I have told, they directly attack on microorganisms because they can form the perforins. These perforins, they contain perforins. Perforins are hole forming proteins on the cell, and once they are able to make a hole, they release the cytotoxic substance into the attacked cell. Then, because of the cytotoxic cells, the cell swells and destroys. Uh, this happens when, if at all, a cell is already infected. Then these killer cells will attack not only the microorganism, the entire cell. Important role in, especially in destroying the cancer cells nowadays, because cancer cells are different, and now uh, in immunotherapy they are using the killer cells. Killer cells will recognize the cancer cell and they will make a hole and then release their cytotoxic substance into the attacked cell only. Only the cells which are infected by the cancer, then they are destroyed. Even in a foreign body or in heart transplant cells, all these things it has been seen. See, the cytotoxic T cells, they will perform, open, and they will release the cytotoxic material here. Then, as a result of the cytotoxic material, cytolysis or cell death takes place. When the cell dies, along with the cell, the pathogen also dies. In our body, there are some professional antigen presenting cells. These are the cells which first find out the antigen and bind with that, and they present them for destruction. Especially the dendritic cells, macrophages, and even B lymphocytes are professional APCs. Uh, Among them, especially the dendritic cells are most efficient antigen presenting cells. They are the main inducer of our primary immune response also. What they do is they present the antigen and activating the negative T cells, and they also express class 1 and class 2 MHC molecule. These dendritic cells are generally found under the skin and mucosa of many organs. So once they cross the skin, then the pathogens come in contact with dendritic cells. They capture the foreign antigen and transport them to the local lymph nodes where there are uh, lymphocytes are there, and they present this antigen to the helper T cells, and helper T cells may develop into uh, killer T cell and destroy. Other cells are macrophages. These macrophages are derived from the myeloid stem cells in bone marrow. They are also produced in the myeloid stem cells in the bone marrow, and they usually are under in circulation in the blood in the form of monocytes or Kupffer cells in liver. And these monocytes, especially the macrophage, they have an important link between the innate and acquired immune system. Innate immune system means immune system that we have inherited by birth. Acquired immune system is in our lifetime we have whatever the immunity that we have developed. Then they are activated and attached to the site of this foreign material by the action of different cytokines and destroy them. This the main function of these macrophages is directly they can engulf and eat phagocytosis. Then osmonization. They will make this antigen inactive. Third thing, these macrophages are antigen presenting cells. They ingest the foreign material, proceed, and the fragment of this antigen are presented to the T cells for interaction. and T cells will help with the help of T cells. They can kill that. Or macrophages may kill the even they kill the antibody coated infected cells also, or tumor cells, because they can also release lytic enzymes, lysis, breakdown enzymes, the macrophages they have. They can kill the infected cells or even the tumor cells also. Uh, they produce different type of proteins and they secrete prostaglandins and synthesize complementary components also. So by all means, the macrophages will try to destroy these cells. See, this is the especially the killer T cells. If the cell is infected or a cancer cell, then bacterial infected cell, the killer T cells terminate the cancer cell and cells infected by virus or bacteria. Especially I am talking about the natural killer cells, the third type of. These are large granular lymphocytes. But on the surface, it lacks the markers like B and T cells. On B and T cells, there are sites, active sites on which the antigens can bind. But natural killer cells, they are also lymphocytes, but they lack the surface markers on B cell and T cells. Usually 5 to 10% of our lymphocytes are these NK cells or natural killer cells. Mainly they are concerned with our innate immunity. They have a spontaneous, non-specific cytotoxic activity. They are not specific. Any pathogen, any antigen, any virus, tumor, graft cells, they will start attacking on them. They are not restricted by either MHC1 protein or MHC2 protein. But it is one thing is sure, MHC class 1 protein inhibit their killing functions. So these natural killer cells undertake the cytolysis like the CTLs, cytotoxic T lymphocytes. Uh, NK cells are non-specific. They are not specific to the antigens. They act spontaneously without prior recognition or activation. And there is no question of MHC protein for the presentation of the antigen. What they do is they destroy the cells coated with antibodies, and this mechanism, the destruction of these cells which are coated with antibodies, it is called as antibody dependent cellular cytotoxicity. Antibody dependent cellular cytotoxicity. Any cell if it is coated already with the antibody means the natural killer cells will kill them. Antibodies produced by the B cells of the immune system recognize foreign antigen and mark them for destruction. And when the antibodies mark any cell for destruction, this is the duty of natural killer cell to destroy them. So during this, the helper T cells already I have told you helps in recognition and identification. Even cytotoxic T cells are also activated by the MHC class 1 or class 2 proteins. Apart from that, there's also one more cells, what is known as suppressor T cells. These are very less known cells. What this suppressor T cell does is, it suppresses the function of cytotoxic and helper T cells, prevent the damage to body itself. Many times what happens, the cytotoxic and helper T cells, uh, if they are not suppressed, they may start autoimmune disorder directly. So they prevent the damage to the body itself. And usually regulatory cells along with the helper cell, they regulate. Thus, they enhances immune tolerance, because you cannot a body cannot go on targeting anything which enters the any foreign protein which enters the body. uh, Usually the cytotoxic cells and helper T cells do like that, but it is the role of suppressor T cells to keep them under control. It's the suppressor T cells, uh, it will instruct the helper T cells, B cells, and macrophages to take rest, not to be overactive. That is the role of these suppressor T cells. And lastly, the memory T cells. These are activated by the antigen but do not enter into circulation. These memory T cells will not be in blood circulation, but they will remain in lymphoid nodes and migrate to various lymphoid tissues throughout the body. These memory T cells attack the same antigen again with immediate response. It will not wait to orchestrate the immune reaction, but immediately it will uh, more powerful and more useful. This is the role of this uh, the memory cells. So with this, we have completed the cells of uh, the immunity, and I will continue with the immunology only for one more class. ಏನು ಕೇಳಿಸ್ತಾ ಆಯ್ತಾ? ಹಲೋ,
Yes, sir.
ಸ್ಲೈಡು ಕೂಡ ಕಾಣಿಸ್ತಾ ಇತ್ತಲ್ಲ? ಓಕೆ, ಥ್ಯಾಂಕ್ ಯು. ವಿ ವಿಲ್
Yes, sir.
ಮೀಟ್ ಇನ್ ದ ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸ್. ನಿಮ್ಗೆ ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸಿಗೆ ಲೇಟ್ ಆಗಿತ್ತು. ಓಕೆ.